Hello everyone, my name is Anna, and my colleague Maria and I have been working on a project to investigate the microbial contamination of cosmetic brushes and the potential use of nanoformulation to reduce this contamination.
Our study was aimed at exploring the types of microorganisms present on cosmetic brushes and assessing the effectiveness of different cleaning methods, including the use of a novel nanoparticle formulations.
We all know how important it is to keep our skin healthy and free from harmful bacteria. However, most of us may not realize that the tools we use to apply our makeup can harbor bacteria and other microorganisms that may cause skin infections and other health issues.
That's right, Anna. With the increasing use of cosmetic brushes, it is essential to understand the types of microorganisms that can be found on these brushes and identify effectiveness of cleaning methods to reduce the risk of contamination. Cosmetic brushes are commonly used for makeup application, but do you know that they can become breeding ground for bacteria?
That's right, Maria. Skin has a diverse range of microorganisms known as skin microbiota, some of which can cause infections.
Identifying and determining the concentration of microorganisms on cosmetic brushes is essential to evaluate their potential health risks.
However, commercially available brush cleaners may not reduce bacterial populations and their effectiveness needs to be evaluated.
That's why we conducted a study to explore the effectiveness of different brush cleaners and identifying a more effective solution.
There is currently a gap in knowledge regarding the types of microorganisms that can be found on cosmetic brushes, as well as the effectiveness of different cleaning methods, especially those using nanoformulations.
Therefore, our study aimed to address this gap in knowledge by exploring the types of microorganisms present on cosmetic brushes and assessing the effectiveness of different cleaning methods, including the novel nanoformulations.
Our hypothesis for this project was that less than 5% of cosmetic brushes contain pathological microorganisms, as well as the microorganisms present on the cosmetic brushes will increase by 10 to 20% for every additional use per week, and 30 to 40% by using products containing oils and waxes.
Commercial cosmetic brush cleaners can reduce bacterial population on brushes by 50% or more and eliminate at least one pathogenic microorganism.
We also believe that over 70% of the microorganisms found in the makeup brushes are also present on the skin or in the cosmetic products used with brush.
Nanoformulations will reduce microbial contamination in cosmetic brushes by up to 50% more than commercial cleaners. To test our hypothesis, we collected a total of 50 hair within the cosmetic brush from the participants and cultured the microorganisms present on them using different agar media.
We then tested different cleaning methods, including commercial cleaners and a novel nanoformulation containing silver nanoparticles, to assess their effectiveness in reducing microbial contamination. Microscopic evaluation was conducted to identify the colonies, and we also tested different microscopic colonies to isolate the expected microorganisms of the skin microbiota. We streaked each plate and incubated them for 24 hours at 35 Celsius to allow the microorganisms to grow.
We also conducted gram staining on selected colonies and reported microscopic descriptions. Moreover, we performed oxidase and catalyst tests on selected colonies to confirm the presence of specific microorganisms. Two users used both for powder and liquid brushes for 30 days. Sampling was conducted every 5 days to monitor the microbial population on the brushes. Brushes were cleaned according to the manufacturer's instructions 1 day prior to each sampling. We followed the same procedure for the same preparation, microbial quantification, and identification for the all samples.
Now, let's move on to the results of our study.
Our findings indicate that the silver nanoparticles infused cleaners were effective in reducing bacterial growth on makeup brushes, particularly those used for liquid products and those that were cleaned after use.
As shown in the figure, the cleaner supplemented with the silver nanoparticles was more effective in reducing bacterial growth on makeup brushes compared to the conventional cleaner. We also found that the use of nanoformulation was more effective with an average improvement of 31% and a maximum improvement of 41%.
The results presented demonstrate that the silver nanoparticle infused cleaner significantly decreased bacterial growth on all types of brushes and cleaning statuses over the 14 days experimental period.
Additionally, we observed that the bacterial growth on brushes used for liquid products, particularly those that underwent cleaning after use, exhibited slightly higher counts than the brushes used for powdered products.
This observation suggests that the composition and residue of liquid products may have a higher uh, possibility to facilitate bacterial growth, and post-use cleaning may not effectively eliminate all bacterial contamination.
The gram staining results for the makeup brushes revealed the presence of different bacterial species. Further testing was conducted using CB plus testing for gram-positive bacilli and STR rapid testing for gram-positive cocci to identify the species. The results revealed that S. constellatus and PET acidilactice were encountered, revealed to the user's history and placement of the brushes.
Only one gram-negative cocci result was received, which is presumed to be Nigeria species due to the nature of the skin-related experiment. The bright yellow colonies observed constantly on TSA plate during the experiment were identified as a Micrococcus luteus based on the gram staining and on a negative oxidase test results.
Our study provides value insights into the types of microorganisms presented on cosmetic brushes and the effectiveness of different cleaning methods, including a novel nano-formulation.
However, there is still much to be explored in this area, including the impact of environmental factors such as temperature and humidity, the effect of high concentration of silver nanoparticles, and the potential transfer of microorganisms from cosmetic brushes to the skin.
We hope that our study will contribute to a better understanding of the risks associated with the use of cosmetic brushes and the development of effective cleaning methods to reduce microbial contamination.
Thank you all for listening, and we would be happy to answer any questions you may have.